Hello, my name is Charles Lindeman, and I am an emeritus faculty in the biology department at Oakland University. I have spent my career studying sperm. Now, when you go to a party or a family gathering, if you're a sperm researcher, people ask you, why would you study sperm? Why don't you study cancer or something important? Hopefully I can convince you that sperm are rather interesting and important. In my paper, I kind of explore the role of sperm in the broader biological world. Now, a spermatozoa has but one function, to carry the genetic material from one generation to the next. They're produced in the haploid phase of the life cycle of eukaryotic metazoan animals, and during that period they have to swim and they have to find the egg and fuse with it. Everyone knows that, but you probably, if you're not in the field of cilia and flagella or sperm, you probably have not heard much about intraflagellar transport. Intraflagellar transport is the system that cells use to build a sperm tail, or to build a somatic cilia, for that matter. And intraflagellar transport, oddly enough, was discovered in a green algae, Chlamydomonas, that has two flagella. Chlammy, as it's called, is the most popular model system for studying cilia and flagella. And a good friend of mine, Bob Bloodgood, was studying how Chlamydomonas flagellis stick together in order for them to mate, and discovered that there is the movement of materials along the surface of the flagellum. Joel Rosenbaum, his mentor, went further and analyzed what was going on at the membrane and discovered intraflagellar transport, which is the movement of rafts of material up and down the flagellum, and it's the system used to build a flagellum. You have to build a flagellum, and IFT, as it's called, is the way that all organisms construct the axoneme, which is the internal scaffold of structures that allows for the production of motility. Now, the IFT system has a fairly large number of proteins and genes in order to be functional, and the sperm tail itself has a very large number of components, each of which have a gene that is responsible for making that protein. And so in the elaborate internal structure of a sperm, one has over 600 different components that have to be assembled, and a very large chunk of genome that has to be working in proper order. The next thing I'd like to introduce you to, if you don't already know it, is that sperm being produced in the haploid phase of the life cycle carry only half of the chromosome number, and they have a very short life, and they're produced in a process called spermatogenesis, which I have a next slide. Next slide shows the steps in spermatogenesis. And one of the things that is remarkable in spermatogenesis is that the first cells to undergo meiotic division to produce the sperm, spermatocytes, they're called, undergo synapsis of the chromosomes. The chromosomes come together, and each chromosome gets at least one crossover. A piece from the other parental chromosome gets swapped out. Some chromosomes will get as many as 4. So all of the chromosomes except for the X and the Y, which are different sizes, get these crossover pieces. And so every spermatocyte is going to produce a unique set of haploid chromosomes that have different crossover patterns. And the two meiotic divisions then produce cells called spermatids that have only half the number of chromosomes. But if they're produced from a different spermatocyte, they'll have a different combination of pieces from the two parental genes. And just recently, just in the last 2 or 3 years, papers have come out and studies have come out showing that there's a big burst of gene transcription in those spermatids that build the sperm, and that a lot of that gene transcription is focused on making the proteins that are necessary for the IFT system and for the building of the flagellum itself, and also for the signaling system that allows the sperm to home in on the egg. The sperm that are produced in the millions are not all identical, all right? And they're actually competing with each other to be the best one that has a good, highly functional flagellum and has a good homing system to find the egg and to fuse with the egg. Right? So this changes the whole view, really, of genetic selection at the cellular level, because if we look at the next slide, right, fertilization success is going to depend entirely on those two factors: whether the sperm can swim well, and on whether the sensory system of the sperm is capable of finding the egg and fusing with the egg. If the sperm is immotile, no genetic material gets passed along. All right. So a good working flagellum and a good working homing system is a gatekeeper to ensure that the genes that are passed along in the flagellar germ-line are in working order, and that they are also the highest quality. They produce the best. Consequently, these genes are identifiable after millions and millions of passages. We have here a picture of 2 modern-day organisms, Chlamydomonas, the green algae, and a human, which I humbly submit is myself in the picture, a younger version of myself. But the fact that you can identify the same proteins and many of the same genes in humans and animals of all kinds, and in Chlamydomonas, the green plant, indicates that there's just an amazing amount of conservation of these genes that participate in this mating ritual. And that means that all of us, every one of us, has an ancestor somewhere way back in the past, at least 700 million years ago, maybe longer, maybe a billion years ago, that was also the common ancestor of modern-day Chlamydomonas. Can you imagine that? we have a great-great-great-grandfather way back there with many greats who gave rise to both us and Climatomolmus. That's pretty, it's something to fascinate and ponder upon, the interrelatedness of the biological world. All right, the last slide shows really the last thing of importance that is a component of the flagellar germ-line hypothesis, and that is that those genes that are so rigorously conserved show up in so many places in the multicellular organisms like humans that it's quite reasonable to suggest that they served as a kind of toolkit for building complex multicellular organisms. All right, to briefly give some example, the rods and cones of the eye are modified flagella. They use a lot of the same genes. The ears, the structure of the hair cells that sense sound and movement are organized by a kinetocilium, which is a flagellar dependent structure. The olfactory system uses cilia as the sensory portion of the cells for olfaction. The brain is loaded with cilia in the ventricles and spinal canal to move cerebrospinal fluid along. And even more remarkably, a large number of sperm genes have been identified as components in building the nervous system. Even organs that you would not think really depend very much on cilia or flagella, like the kidney, it turns out that they depend on a flagellar-like structure called the primary cilium that is present on almost all cells in the body. And the primary cilium plays an extremely important role in embryonic development. All right. During embryonic development, the primary cilia on the cells in the embryo are the sensors, the antenna, to sense the developmental factors that are released and to determine the differentiation of the cells into different tissues and organs. So literally, the genes from the sperm tail and the sperm sensory system have been used in the process of forming embryonically developed multicellular organisms. Put it all together, and maybe you can see now why sperm are maybe more important than we all thought. And that they possibly have played a major role in the development of complex multicellular organisms and the developmental process. If you read my paper, you'll see that there are many more examples to support this idea. And in the last slide, I put together a little picture there showing that the competition between sperm may have played a role in the development of all of the metazoan organisms. And interestingly enough, that may have contributed to making you and me possible. Thank you for your attention. I hope you will go on to read the paper and think about it yourself, and maybe it will stimulate research to see if this concept has validity. Thank you.